chapter 21, genomes and their evolution. So, evolutionary history. As we see here, this is a chimpanzee. It's our closest living and relative in evolutionary history chart. This is a current day human. But what accounts for these genetic changes in evolutionary history? Well, now that we've been studying genome sequencing, we're getting closer to finding out exactly where the differences come along. So go over a presentation, there's 4 steps. We're going to discuss the 2 different approaches to genome sequencing. We're going to summarize what has been learned from genome sequencing, describe the composition of a human genome, and then talk about exactly how they evolve. A few basic terms you should know. A genome is a genetic material of an organism, pretty much all the genes. Genomics is a study of the whole sets of genes, in other words, the study of the genome. And bioinformatics, the application of computational methods to store and analyze the data. Bioinformatics is very useful, especially in terms of studying genomes because of the plethora of information there is. So in 1990, the Human Genome Project began. It was virtually completed in 2003. Basically, the goal of it was to map the entire human genome. When they say mapping it, it's basically determining the complete nucleotide sequence. So over time, breakthroughs in technology obviously sped up the process. For example, originally we only had 1,000 base pairs a day, 1990s, but now, 10 years later, they can do 1,000 base pairs a second. So originally the genome approach to sequencing was slow, tedious. 1998, J. Craig Venter came up with the whole genome shotgun approach. And instead of reading, I'll just show you a picture. So they basically had the DNA segments here. They cut it up into small overlapping fragments. For example, they had nucleotide sequences, but these would overlap. And then a computer would automatically file them all into one continuous sequence and thus find new protein sequences faster. Alright, so metagenomics. It's from the Greek word meta, meaning beyond. They collect the DNA from an entire group of species, from a single environmental sample, and then they sequence that. And a computer then accomplishes the task of sorting out the partial sequences and assembles them into individual specific genomes. Metagenomics is used in microbial communities, such as the human intestine. The ability to sequence this DNA in mixed populations eliminates the need to culture each of the species separately in a lab, thus speeding up the process. So 21.2, bioinformatics. As data began to accumulate, they needed to start working together to finally put all these sequences together. Through the coordination between scientists and the government overseeing the Human Genome Project, a database and software were set up all around the world. They even made all this data available on the internet for everyone to post and pull out information. The government combined the National Library of Medicine and the National Institutes of Health and created the National Center for Biotechnological Information. The NCBI,
What does HGB stand for?
Human Genome Project. The NCBI has a website, or has a database called GenBank, and on this has every single nucleotide ever sequenced. And so any scientist, if they need to access some, they can go look at it and pull out information that they need to help them sequence theirs. They have a special software called BLAST, which allows the wizard to compare DNA sequence, so like I just pretty much said. They can also compare predicted protein sequences, and they can search for any protein sequence among stretches of amino acids. Alright, so to identify protein coding genes, so gene annotation, analysis of genomic sequences to identify protein coding genes and determine the function of the product. Now what does this mean exactly in layman's terms? Well, so basically what do the scientists do with these long stretches of DNA sequences? Basically they're trying to identify all of the protein coding genes and their functions. That's their main goal. So basically what the computer does, the computer scans for transcriptional and translational start and stop signals in RNA splicing sites. Does anyone remember the start signal for transcription?
Like CNT.
I'm looking for the amino acid start part.
Or something?
Yeah, methionine.
Yeah, methionine.
AUG. Some stop signals include UAA, and UAG. They also look for sequences to specify mRNA, which are called express sequence tags, or ESTs.
Okay, so understanding genes and gene expression. If you recall from earlier, genomics is the study of whole sets of genes and their interactions. And it is also a rich source of new insights into fundamental questions about genome organization, the regulation of gene expression, as well as their growth and evolution. A program was started in 2003 called ENCODE, and this stood for the Encyclopedia of DNA Elements. And here, researchers focused on 1% of the human genome, and their goal was to learn all they could about the important elements in that certain sequence. And in the program, they looked for protein-coding genes and genes for non-coding RNAs, as well as sequences that regulate DNA replication, gene expression, and chromatin modifications. The project was hugely successful and led to 2 more studies. One continued the analysis of the human genome, and the other used the soil nematode, Chironahabdus elegans, I believe, and the fruit fly Drosophila melanogaster. And they were both used as these species could be experimented on with genetic and other biological experiments. And because of this, testing the activities of potentially functional DNA elements in their genomes will reveal about how our genome works. And the success with genomes led to the study of protein sets, which is called proteomics. And these proteins carry out most of the activities in the cell. So when we study the proteins in an organism, we can further understand how these proteins interact in networks and the function of cells and organisms much better. And genomics is allowing biologists to study life at the global perspective. We can study different parts of an organism, and this leads to and how their cells operate and how their function is integrated into biological systems. So one important use is to define gene circuits and protein interaction networks, and this can be has been done in a yeast called Saccharomyces cerevisiae. And what scientists can do in this specific yeast is they can disable 2 genes and create what's called a double mutant cells, and they can compare how these cells function to the regular cells in the yeast, and then from there they can figure out what gene products interact with each other and which ones do not. And here is an example of like systems biology. The computer software is able to map these genes into like a functional map that is able to process the vast number of protein-to-protein interactions. And this takes highly advanced technology, and is just becoming more useful because we didn't have the technology like 10 years ago. So in medicine, systems biology has been applied to the modern world in many ways. An example would be the Cancer Genome Atlas. And this project was headed by the National Cancer Institute and is trying to determine how changes in biological systems can lead to cancer. And this project was 3 years, and it began in 2007. And it set out to find all the common mutations in 3 types of cancer, which were lung, ovarian, and glioblastoma of the brain. And they did this by comparing the gene sequences and patterns of gene expression in cancer cells to normal cells. And this has also been extended to 3 other common types of cancer and may lead to new types of therapy and treatment. And it also has tremendous potential in the medical field, as silicon and glass chips have been developed and have the ability to analyze gene expression patterns in people suffering from cancer and other diseases. And this could, again, lead to specific tailoring of a treatment for that certain person based on how your genes are made up. And by early 2010, the sequencing of about 1,200 genomes had been completed, and 5,500 genomes and 200 metagenomes were currently in progress. And this is just obviously a wealth of information that's only been, the surface has only been scratched, so there's much more we have to learn. Okay, so genome size. When you compare the three genomes from archaea, bacteria, and then multicellular eukaryotes, there's a general difference in size. However, the genomes of archaea are within the size range of bacterial genomes, and eukaryotic genomes tend to be larger. For example, a yeast cell has about 12 million base pairs in their genome, where a multicellular organism has about 100 million base pairs. And a study comparing phenotype with genotype failed to reveal any systematic relationship between genomic size and an organism's phenotype, so that really has no effect on the phenotype. And here's just a table comparing some of the genome sizes. So humans, we have about 3 billion base pairs, whereas if you compare it to like a bacteria, they only have 4.6 million, as we're much more complex organisms. And like I said, the number of genes also varies between prokaryotes and eukaryotes. And bacteria and archaea, since they're much simpler organisms, tend to have fewer genes than eukaryotes. Whereas bacteria have about 1,500 to 4,000 genes. Multicellular eukaryotes can have up to 40,000. And after scientists discovered that we had 3 billion base pairs, they expected that we were going to have about 50,000 to 100,000 genes. But they discovered that we only had about 21,000, which is a very low number. And it could be comparable to things like nematodes, which are very simple organisms. And they found this was a result in evolution, as it was efficiency in multicellular organisms, because RNA can code for these genes for multiple proteins. And this gives us an advantage over other organisms. And this additional polypeptide diversity could result from post-translational modifications, such as cleavage, or the addition of carbohydrate groups in different cells types or at developmental stages. So this is just a result of evolution over time, how we're able to more efficiently code for DNA. And we can also measure the gene density. And even though we have many more genes than bacteria, they have a higher gene density because we are multicellular and have hundreds of millions of times the base pairs. And we also have approximately 10,000 times as much non-coding DNA as bacteria, and these are called introns. And if you remember, introns are the non-coding sections of the RNA transcript. And these account for the differences in average lengths between the human genes and bacterial genes.
So mostly we've been just talking about genes that code for proteins, but the coding regions for these genes and the genes for non-coding RNA products such as RNA, tRNA, and mRNA only make up really a small portion of the genomes of most multicellular eukaryotes, and I'll have like a graph for that in a second. Only about 1.5% of the human genome codes for proteins or into like rRNA or tRNA. Gene-related regulatory sequences code for about 5%, and then the introns account for about 20%. The rest is located between functional genes. And you can see this graph, it's like the one in the book. You can see that the introns and regulatory sequences are about 24%, and really the exons, the ones that code for like the proteins, are only about 1.5%, and the rest is all these different types.